Myself, Anagha, Assistant Professor, Department of Microbiology, KSR College of Arts and Science for Women, Tirchankot. And I'm going to talk on the topic DNA repair mechanism, photoreactivation. DNA damage refers to the change or disruption that occurs in the DNA molecule caused by either environmental factor or normal procedures that happens in our body, in our cells. And the types of damage include DNA strand breaks, oxidative damage, alkylation of bases, base loss, bulky adduct formation, and DNA cross-linking. The sources of DNA damage can be either exogenous or endogenous. Exogenous DNA damage sources include replication error, topoisomerase activity, reactive oxygen species, alkylating agents. And the exogenous DNA damage include ionizing radiation, ultraviolet radiation, exogenous alkylating agents, aromatic amines, and PAH. To cope up with the DNA repair, DNA damage, our body, our system consists of a repair mechanism, lots of repair mechanism, and photoreactivation is one of that. Photoreactivation repair is the DNA repair system which corrects the damage caused due to UV rays, that is UVC and UVB, with the help of photolyase enzyme. Photoreactivation is also known as light repair, as the photoreactivation repair is a unique method that repairs the DNA damage induced due to a light with the help of a light itself. And it is also known as direct reversal DNA repair mechanism. And this repair mechanism can occur both in prokaryotes and eukaryotes. The photoreactivation process repairs the UV damaged DNA, and the DNA helix becomes distorted and forms a pyrimidine dimer, most commonly thymine-thymine dimer, under the UV light exposure. A dimer forms when the adjacent thymine bases attach covalently to their respective C5 and C6 atoms. A formation of thymine-thymine dimer causes desorption of the helix, which eventually causes replication error in the DNA and leads to the mutation. The covalent bond formed between the two adjacent thymine bases must be broken to recover the desorption. An enzyme known as photolyase can break the covalent bond formed in the dimer. And this picture shows the process. Actually, when UV light is exposed, a dimer is formed, and this leads to a formation of a bubble, and this bubble can be cleaved with the help of photolyase enzyme in the presence of visible light, and thus the structure of the DNA is regained. Mechanism of photoreactivation. The enzyme photolyase consists of two cofactors, FADH and MTHF. FADH flavin adenine dinucleotide and MTHF is methylene tetrahydrofolate. So initially the MTHF will absorb high energy wavelength light from the visible light, that is most probably 384 nanometer, to repair the DNA damage. When it absorbs the energy, it will get excited, and the excitation of the MTHF molecules will transduce photons or electrons to FAD molecules. FAD molecules accept the electrons and will be getting converted into FADH2 molecules. The FADH2, which is now a high energy molecule, can get attached to the thymine dimer, and after binding to the dimer, it will break the double covalent bond between the cyclobutane thymine bases, and thus the photolyase enzyme will reverse the cyclobutane pyrimidine bases to usual base of DNA via photochemical reaction. Steps included. First step is the formation of the pyrimidine dimer. When the pyrimidine dimer is exposed, pyrimidine basis is exposed to UV rays, a dimer formation will occur, that is the two adjacent pyrimidine bases bind through a carbon-carbon double bond, that is a covalent bond formation will occur, and it is referred to as cyclobutane pyrimidine dimer. Second step is a treatment with photolyase under visible light. When the pyrimidine dimer is formed, a bubble formation will occur, and the photolyase enzyme under the visible light of a longer wavelength will scan after getting activated. After scanning, it will recognize the pyrimidine dimer, and then it will leads to the binding of the photolyase enzyme to the segment directly. Breaking of thymine dimer. When the photolyase enzyme binds to the dimer, it will start splitting the carbon-carbon double covalent bond between the cyclobutene rings, and thus it will convert the dimeric form of pyrimidine bases into monomeric form. After the process of breaking the covalent bond, it will be getting released, and thus the DNA get fixed. That is the last step, that is the release of photolyase. And thus, with the help of photolyase enzyme, the damage caused due to the UV rays can be fixed. Thank you. I hope this video is helpful to you.